this academic ceremony in which Mr. Al Jowf will defend his academic thesis, Biomedical and Public Health Studies on Susceptibility to Post-Traumatic Stress Disorder. I welcome the candidate and wish him lots of success. I also welcome the supervisors, Professor Rutten, Dr. Eijs and Dr. De Nijs, and all family and friends, and of course, the members of the opposition whom you will meet later. But first, I invite you to give a short presentation on what you have presented in your thesis. Mr. Candidates, the floor is yours.
Go ahead, Ghazi.
Hi.
Yeah, you can start your presentation. I welcome you, wish you success, etc. But you can start.
I think I believe you are able to see it, right?
That's the wrong screen, Ghazi. That's presenter mode.
Sorry about that.
That's correct.
Yeah. Sorry about that. Dear Proctor, members of the Corona, supervisors, colleagues, family, and friends. In the next 15 minutes, I will present my PhD research entitled Biomedical and Public Health Studies on Susceptibility to Post-Traumatic Stress Disorder, abbreviated as PTSD. People might be exposed to shocking event in their lives, which can be a scary or dangerous experience that can affect someone emotionally and physically. Such traumatic events can lead to a condition called post-traumatic stress disorder, abbreviated as PTSD. Examples of traumatic events are those caused by human behavior, such as combat and assault, as well as by nature, like natural disaster. There are 4 symptoms clusters that characterize PTSD, which are avoidance to stimuli, such as reminders of that traumatic event, intrusion, such as flashbacks and nightmares, arousal, such as irritability, and lastly, the negative emotions, such as feeling of guilt. The general scheme of PTSD development could be illustrated in a simple way. When somebody is exposed to traumatic stress, an acute stress response is initiated. Thereafter, either the person is resilient, so after their initial stress response, they recover, or they are susceptible, so their stress response culminates in the development of PTSD. This has led to the recognition of the inter-individual susceptibility to PTSD. This susceptibility is dependent on many factors, such as biological factors like genetics, chemical changes, or organ damage. Also, psychological factors, including stress, mental illness, behavior, and personality. And lastly, social factors such as peers, socioeconomic status, beliefs, and culture. In my thesis, I took 2 perspectives, one to public health and the other one to molecular biological studies. The first aim was to address the gaps of knowledge in the molecular mechanisms determining susceptibility to PTSD. The second aim was to identify the public health measures that can be effectuated from the prevention of PTSD and the utility of molecular biomarkers. I will first focus on presenting the work I did for aim 2, which consisted on literature review on public health of PTSD. To understand how PTSD can be viewed from a public health perspective, one can use the socio-ecological model as a looking glass. It is a model of stepwise prevention approach, with the individual being the core of the model, as one would expect, followed by an individual relationship also their community, and lastly, the larger society. Specific prevention methods relating to the individual can be applied at different levels to allow for better prevention. Once traumatic experience occurs, very traumatic factors play a role in mediating the response besides the type of severity of the traumatic event. As we already saw, these can be biological, psychological, and social factors, and they can play at different levels from individuals to society. Together, these factors determine whether after acute stress response, the individual recovers or develops PTSD. However, before we experience a trauma, there are factors that define your susceptibility called peri-traumatic factors. These are, for example, early lifetime experiences as well as genetic factors. Knowing these factors, preventive strategy can be implemented. There are three main types of prevention. The primary prevention, which takes place before the person is exposed to trauma, aims to prevent the onset of illness and prevent a traumatic response. The secondary prevention takes place right after traumatic exposure and aims to alleviate the response and the complications of condition. The tertiary prevention takes place after the development of disorder and aims to control the condition and prevent further irritation. In order to probably implement these preventive strategies, biomarker can be used at different stages. As seen, biomarkers allow timely prevention and treatment starting even before someone is exposed to the traumatic stimulus and until treatment is established. Since there are various treatments for PTSD, including, for example, psychotherapy and the pharmacotherapy, therapeutic biomarkers can help select the best strategies for individual patients. Let's now discuss a bit what biomarkers are and how they work. Biomarkers is a measurable characteristic that can be a substance, responses, or structures used in an indicator of pathological process. Examples of biomarkers could be brain scan, blood measurement, or something like urine or saliva. In other study, the main focus was on molecular biomarkers. Of molecular biomarker, the detection of the activity of genes are very important. Nowadays, we have technologies to measure activities of many genes at the same time. Activity of a gene is regulated by a process called epigenetics, which correspond to an addition of a little switch to a DNA, which determine whether gene turn on or turn off. These epigenetic mechanism can be detected by specimens taken from the person, such as urine, stool, blood, sputum, or even cerebrospinal fluid. This makes epigenetic mechanisms good candidate biomarkers of disease, in particular PTSD, which I investigated in the first aim of my thesis. As already mentioned, the aim is to address the gaps of knowledge and the molecular mechanisms determining susceptibility to PTSD. To specifically investigate, in chapter 3 and 4 of my thesis, I conducted 2 studies on some of the genes that are thought to modulate the stress response and are regulated by such epigenetic mechanisms. I will start presenting the work done in chapter 3, and for that, I first need to introduce the PRISMO study. PRISMO is a prospecting research and stress-related military operation. It is an extensive cohort of more than 1,000 Dutch soldiers assigned to a 4-month deployment to Afghanistan between 2005 and 2008 that has been followed up over time from before deployment till 10 years after. From a subset of around 40 male soldiers, we had blood samples taken at 6 months after deployment. Based on the psychological evaluation, we were able to divide the soldiers into 2 groups, resilient and susceptible. And in these blood samples, we actually measured how much gene were turned on or turned off. This activity measurement we combined with data on the epigenetics regulation that we had generated before from the same individuals. We found that 21 genes for which their activity in blood was different in individuals susceptible to PTSD. Several of these are involved in non-relevant stress-related process related also to the immune system and cellular signaling mechanisms. Genes found include FOSP, for example, and TBC1, D16, which are of interest, as the first was previously well studied to be activated in the state of the chronic stress. And the second, of course, offers a new angle to genetic factors involved in PTSD susceptibility. As a conclusion of our PRISM study, we established potential links between gene activity and PTSD susceptibility. Our results add to the understanding of genetic factors and biological mechanisms that are involved in PTSD. Now I will continue by explaining the work which is done in chapter 4 on the RNA-39 study on mouse brain samples. From a previous epigenetic study done in our laboratory at Maastricht University using a PRISM cohort, we had identified RNA-39 as a candidate gene associated with PTSD that may serve as a biomarker. However, first we had to validate the possible function of RNA-39 in the brain and whether if there are any changes in PTSD. In order to explain our work, I need to introduce the brain regions involved in PTSD. This is a picture of a cross-section of the human brain, and here you can see some of the regions involved. The main part related to my study is the hippocampus, which is the main important region for consideration and maintenance of memory. But our study, we did it only on by using human brains that we used mice brains instead, obtained from the study done previously in our laboratory. We used a mouse model of PTSD in which we exposed a mouse to severe stress by continuous exposure to another dominant and aggressive mouse for 15 consecutive days. These mice were compared to unexposed control mice. We used the brain of those animals to analyze the quantity or the amount of RNA-39 in the PTSD regions, and I will show you the result of the hippocampus. First, we looked where the RNA-39 protein was located in hippocampus, which is divided into 3 regions, CA1, CA2, 3, and the DG. We colored brain sections from mice to see where RNA-39 is located, and the more RNA-39 is present, the darker the color we get. We find that the RNA-39 is located in those regions, and its distribution looks a bit like fibers. From these images taken from the microscope, we measured the color differences between the different groups in the 3 hippocampal regions. Then we found no difference in the quantity of the RNF between unexposed control mice and mice exposed to severe stress. In addition, we measured the length of RNA-39 fiber, which was clearly increased in the mice exposed to severe stress in DG brain regions, as compared to the unexposed, of course, control mice. And now turning on the result of our RNA-39 study, considering the most important findings related to the PTSD, we confirmed that RNA-39 is present in the hippocampus and that changes are present in mice exposed to severe stress as compared to unexposed control mice. Our findings supported the role of RNA-39 as a potential mediators of stress susceptibility. Now I discuss with you all the chapters of my thesis. First, I started off with the general merits of biomarkers and the potential of epigenetic biomarkers. Then I went into dedicated experiment studies that, as we already mentioned. That completes my overview, and now I will sum up the main conclusion. I concluded that the integration of medical research and public health forces can result in significant reduction of disease burden, especially in the diseases or in disorders like PTSD. Further research on PTSD susceptibility is needed to derive cost-effective biomarkers that can be used on a public health basis. Herewith, I complete my presentation. I thank all for your attention, and I give back the word to the professor.
Thank you very much, Mr. Candidate, for a clear presentation. We will now start the opposition, and the first opponent is also the chairman of the assessment committee, that's Professor van den Hove, and he is professor in neuroepigenetics in the Department of Psychiatry and Neuropsychology in the University of Maastricht. Professor van der Hoof.
Thank you very much, Mr. Pro-Rector. First of all, dear candidate, my congratulations and compliments for your thesis. I think with a public health background, diving into biology must not have been easy, but you made it. So my compliments there, and also my congratulations and compliments to your team of supervisors. Of course, first some questions, and I first want to start with a general question. Also compliments, by the way, on your presentation. A general question which you touch upon for the first time in chapter 2, but also at other occasions in the thesis, where you state that knowledge on various layers of epigenetic regulation and its role in PTSD, of course, will help in the development of biomarkers, but also in the development of better treatment strategies. So generally, more insight there will be of great help. And you give some examples throughout the thesis, looking in the blood, for example, like you did at methylation. You can also look at postmortem brain tissue, comparing patients and controls, and you also use animal models to get more insight. Now, I was wondering in general, how do you then assess these and interpret these associations? Because whatever you do, all those options that I just described will give you an association between an epigenetic process and in this case, PTSD. So how would you study causality to see whether it's more than just an association? Because some of these observations might also represent the consequence of the disorder and not necessarily be representing biological processes involved in its development. Could you share your thoughts on this?
Yeah. Highly esteemed opponent. First of all, thank you for your compliment and your question. Regarding to your question, I believe in many cases we could find like an association between, for example, like a disorder or a disease, and for example, certain genes that are available in the human body, for example, or an animal. But the main issue is when and where this could be, for example, active or not. That's, for example, one thing. If they are, for example, available and present and they are active, so the association, for example, could be possibly drawn if that's the case. But in terms of, for example, the causality, sometimes some DNA, for example, methylation can, for example, mean to or direct you to a specific causality thing, which you can maybe think this certain disorder or disease could be not only drawn only from such, for example, samples or like biomarkers that you could find in their, for example, tissue or cells. So I believe
I agree. I understand. So, for example, let's look at RNF39 there. Let's say you would even collect more evidence on changes between stressed animals, control animals, or maybe PTSD brains versus control. Let's say you gather more evidence. How could you really then prove that regulation of RNF39, either at the level of methylation, but it could also be in general, is involved in the development and course of PTSD? How could you try to tackle that, for example, in an animal study?
Yeah, of course, in our study related to the RNF39, we came up to, for example, the first result that we saw, for example, there is, let's say, there is a difference, for example, in fiber length, for example, in hippocampus regions that could be sure that there is, for example, RNF39 gene present in the fibers, and the length of, for example, of that fibers themselves changes. Also, we cannot say, for example, this exact, for example, part in the cell of these genes, for example, were present or available. So it's a little bit difficult sometimes to come up with a presence, for example, of a particular gene in a cell or like in a tissue, and come up with a conclusion that by saying this is like a good evidence or a lead to a particular, for example, disorder such as, for example, PTSD. But with having so many sample size, for example, and drugging into more research and investigation, and by looking into, for example, the cell body itself and the genes that they are active or not active, and treating or comparing between, for example, the control and social defeat, for example, animals or exposed animals.
I'm going to interrupt you.
To stress might be.
Yeah, you're heading in the right direction, and this is all also partially stuff you already did, so that's nice. What about epigenetic editing? If you would epigenetically edit RNF39, could you think of an experiment in cells or in animals that would then prove causality?
Yeah, in terms of epigenetic editing, we haven't, of course, went through such this procedure in particular. It wasn't part of our investigation or research in particular, but I believe it could be a possibility, or there is a possibility. Such things might also increase the evidence by having, let's say, a biomarker or an activity of a particular gene in that, for example, animal or human being tissue. So it could also cause at the end what we call it a particular evidence which come up together by changing, of course, the affiliation of having such, for example, a disorder or a disease.
Okay. Let me move to chapter 4, and actually we already touched upon that a little bit by talking about RNF39.
Yeah.
One thing, and I think you touch upon that yourself as well, the first finding, for example, on page 103, that I think is of great interest, well, it's not the first one, the first one I want to discuss, is the change in total RNF39 fiber length, and you also actually showed it in your presentation.
Yeah.
To which extent do you think this finding is specific? Because it could also be that there's generally more fibers, or the fiber length is increased after social defeat, and that RNF39 is just a bystander or just an epiphenomenon that's increased. To which extent do you think you could tackle that? So to be sure that it is actually RNF39.
Very short answer, please. Very short answer, Mr. Candidate.
Yeah. Yeah, of course, we cannot say which exact part in the cell these changes were, but could be, for example, an example in dendrites or in the axons. But we try to investigate. Also, we need to stain and have further cellular, for example, markers. And of course, our laboratory in Maastricht are trying, of course, in the meantime, to process further investigation in terms of having further staining in, for example, the particular, for example, region of the hippocampus, which is the DG in particular, for example, to be able to tell if, for example, those or this, for example, RNF39 is fully present. But in terms, of course, of the fiber length, we find that there is an increase in fiber length, of course, after for those animals who are exposed to traumatic stress.
Okay, thank you very much. I give the word back to the program.
Thank you, Professor van der Hoven. Then we come to the second opponent, who's also a member of the assessment committee. That's Professor Steinbusch. He's Professor of Cellular and Translational Neuroscience in the Department of Psychiatry and Neuropsychology in the University of Maastricht. And he has also accepted the role of secretary today, for which I'm grateful. The floor is yours.
Thank you, Mr. Pro-Rector, and I'm also pleased to join this committee. I would like to congratulate the candidate. I had the opportunity to meet him about a month ago, in which we have, or 2 months ago, I think, in which we have an in-depth discussion about his thesis. And I also would like to congratulate the full promotion team in which he has worked with. My questions are related, and I will continue the opposition which was started by Professor van den Hoven. And I also move a little bit to chapter 4. And of course, I was invited to this event, so you will get some neuroanatomical questions related to your work. So first of all, Mr. Candidate, you mentioned on page, and I'm following a little bit, on page 10, that PTSD is basically, I would call it, and I push you a little bit, a female thing. You mentioned there on your chapter says that this postnatal stress disorder is twice as much available in women than in men, and it's much more available also in young adults. So my first question is, when you conceptually were working with your team, with Professor Rutten and the team on your chapter 4, you decided to use male rats. And I assume, I give you already a little answer to that, I assume because they were just there. But would you think later it would have been better to move to female rats? Can you give me a little bit of opinion on that?
Yeah, a highly esteemed opponent, thanks for your question and of course your compliment. Yeah, regarding having a male, for example, mice in our laboratory, and we used, of course, as you know, a previous study which has been done in our, of course, institute. So we took the advantage of getting tissues or samples which we were, or they were actually investigated earlier. So basically what we had is in general like male mice. However, I would love, of course, maybe later in the future, if we could maybe add further stainings or analysis to our study, of course, by adding female mice, just to see if there is any differences across those two gender.
I would like to continue, and I also take then your nice presentation in which you showed there was a strong emphasis in your work on the hippocampus, and you also mentioned in the amygdala complex. And then you mentioned page 96, yes, 96, that you found shrinkage strong shrinkage in the volume of the hippocampus. So my first idea was, if you see something in the hippocampus appearing, and you're going to do behavioral testing, you should also have a test which is measuring this, a memory task. But the 3 behavioral tests you're using, they're only related to anxiety-related behavior. So why didn't you add anything which measures the changes in your hippocampus in the behavioral aspect?
Yeah. Frankly speaking, as mentioned earlier, of course, the behavioral test has been done previously. So I wasn't able, of course, to, or I wasn't part of that study in particular. However, they already applied those behavioral tests, and they came across, of course, for example, the social interaction test for the evaluation of social behavior. And this, for example, followed on other tests. But mainly, they try to investigate whether if there is a general, let's say, trend or which could lead us to going or to investigate or to come up across having such, for example, RNF39 protein in those, and the association of course, being or getting a PTSD. Because also with other studies, there was, of course, a clear association for, especially in particular, for patients who diagnosed as PTSDs, and they came across also having or finding the RNF39.
I just interrupt because the time is limited, and I look to the pro-rector if there is time for a short question.
Very, very short question and a very, very short answer.
No, then I just leave it. I don't. If it's getting too short, I see here it's 30 seconds for remaining. Makes no sense. I complete my opposition, and I give back the word to the pro-rector.
Thank you, Professor Steinbooth, and thank you for helping us keeping the time. Then we come to the third opponent, and is also a member of the assessment committee. That's Dr. Van Mierlo, and he's associate professor of neuroscience in the Department of Psychiatry and Neuropsychiatry, Psychology in the University of Maastricht. Dr. Van Mierlo.
Thank you, Mr. Pro-Rector. I would like to start to congratulate the candidate and the promotion team with the thesis. As already being said, this thesis merges knowledge in the public health domain with hardcore neurobiology, which is not an easy task. So hence my compliments for merging that knowledge. Nevertheless, I would like to discuss some concepts that have been put forward in the thesis. And I would like also to discuss some follow-up experiments with the candidate. So my first question relates to your review in chapter 2, and you refer to an increase in brain-derived neurotrophic factor, which was detected in the plasma of PTSD patients, and also an increase in BNF, which was detected in the inescapable tail shock test in rats. So personally, I would expect that BNF signaling would be beneficial to PTSD patients, since an increase in BNF could contribute to neuroplasticity and has been suggested to be a treatment target even in depressive-like disorders. So according to you, is BDNF merely a biomarker, or is it a potential contributor in PTSD neurobiology?
Esteemed opponent, thank you for the question and of course for your compliment. The BNF in particular, of course, it's part of the biochemical mediators, which could lead or to give us a hint about, for example, PTSD in general, if we had it or not. So it's also this also, or this in particular, it's not, or it doesn't play only a role. Also, like the, for example, the MBY and the GABA in particular, they also play a specific, for example, like role for mediating or to increase, for example, the having the PTSD in general. So if you want, for example, so we consider it as a biochemical, of course, factors which may introduce us to getting or having like PTSD.
Could you perhaps speculate on the potential biological role BNF might play in this context?
Sorry, if you could repeat your question, I didn't understand.
Well, could you perhaps speculate on the biological role of BDNF in this context? So why would it be? Can you give an explanation why it's upregulated after PTSD?
The only thing that with patients who had PTSD, the symptoms, for example, are not giving us, for example, like a hint, which they can be, for example, diagnosed or like mentioned as PTSD patients, or in particular. So the linkage, it's not, of course, that easy, but with many biochemical, for example, like mediators, all together, we could come up, for example, with the result that those, for example, upregulating or downregulating some particular, for example, genes to investigate the need, of course, of having or knowing the PTSD in general.
Okay, thank you for your answer. I'll take it to next target, which you addressed in chapter 2. You nicely distillate that related to PTSD, one of the most consistent differentially methylated CpG sites across the multiple studies is happening in the DOC2 region, so in the DOC2 gene. And DOC2 is coding for a protein involved in intracellular signal networks, mainly in leukocytes, but in the central nervous system, also in oligodendrocytes. Could you speculate about a potential functional involvement of DOC2 in disease pathology?
Yeah, of course, we haven't studied this in particular, but So I'm not quite sure what, for example, tend or lead could take us to, but I believe such, for example, like biochemical, for example, mediators, of course, might play a role in general, and combining those together could, of course, lead us to get kind of like a result, or to have an idea what's really happening in there, and how can we differentiate, for example, between people, for example, having PTSD, but the people, for example, the main focus was on investigating the susceptible and resilient patient in particular.
And so more to the molecular level and to the cellular interactions, since DOC2 is expressed in leukocytes and is involved in phagocytosis, do you think it might contribute by affecting the synaptic densities in the central nervous system? Would this be a potential role of this DOC2 protein?
Yeah, since we haven't fully investigated this in particular, so I cannot really tell what is exactly really happening. But based on our investigation or like previous studies that we have done, certain, for example, like mediators might play a big role in terms of knowing further, for example, like scores or scales that we might get to be able to differentiate and divide our group, for example, in general.
Okay, thank you. One last question. In chapter 3, sorry, in chapter 4, you in chapter 3 it is, sorry. You study 20 susceptible and 20 resilient PTSD samples, and you also describe some limitations, like the sample size used, but also the non-FDR corrected nominal p-values, and perhaps an overinterpretation of the GO analysis. So if you would have to describe an optimal study design, how would you approach this? It can be very brief.
First, of course, in most studies there are, I believe, many limitations. You cannot go across everything at once. So you have to investigate and of course, try your best to come up with limited limitations, let's say it this way. But in that study in particular, for example, one of the limitations was also having like only male surgeons in particular. It's similar to the our study, of course, in chapter 4 related to the RNF39. Also, there was concepts, of course, of white male, of course, Caucasian individual in particular. So we haven't studied different, for example, ethnicity or so. Also, there is an observing of changes over a longer period of time. So we haven't also like took in consideration the 5 different time points that we had. Okay, we only came across.
I think we can continue this discussion on the next occasion. I'm satisfied with your answers, and I close my opposition here due to the sake of time. So I give back the word to Mr. Pro. Thank you.
Thank you, Dr. Familo, for your position. Now we come to the following opponent, who's also a member of the assessment committee. That's Professor Youssef. He's Professor of Psychiatry and Behavioral Health in the Harding Hospital in Columbus, USA, Department of Psychiatry and Behavioral Health in the Ohio State University. And thank you coming all the way from Ohio, albeit online, because it's about 7:45 in your country. The floor is yours.
Thank you, Mr. Pro-Rector, and congratulations, Mr. Jowf. Congratulations to the full promotion committee, and it's an honor to be here with all the committee and the assessment committee. And I would like to start by asking a couple of general questions. We go to a specific question. So, an overview. What would you think of, or speculate even of the biggest effect size of all the biomarkers for PTSD, or what would be the most important one I would think the field moving forward should target? I know, and I agree with you that all are very important, all of them together contribute, but what would do you have in your work over the last several years? Have something stood out as one of the biggest high yield biomarkers?
Highly esteemed opponent, many thanks for your question, and of course for your compliments. Yeah, there are different biomarkers, as you know, and sometimes we pick up, for example, like a particular one, like for example, blood, to maybe, let's say, certain studies, but not because, for example, we are only interested in such biomarker, but of course, it limits us sometimes to the sample availability. So it's not really an easy to came across different biomarkers for the same studies or investigation. For example, in our Prismo, for example, study, we do had urine and blood, for example, samples, but we couldn't, for example, process, or in my case, I couldn't or wasn't possibly process anything apart from, for example, blood. So it's difficult to say there is one particular biomarker which we can go with in general. I believe in most cases the best is to have as many as possible to be able to know and duplicate, for example, our investigation or studies to be able to see whether we have the same result or the outcome.
Yeah, so like there's nothing stand out whether some neurotransmitter, neuropeptide Y, or some epigenetic markers. You're saying that several of them are important, and there's no one that has a bigger effect size than the others. Is that correct?
I would say, like in general, yes, if we could have all of them together, but of course this limit to us to the timing and of course the sample size and so on. So it depends.
Okay. So I would ask regarding a public health effort to either prevent or treat PTSD, what would be after your studies and your PhD, what would be the biggest thing you think would be important to implement? Again, there is several measures I realize and stuff, but what's really an important thing that at least the field should study more?
Yeah, first of all, we have to take in consideration that there is no specific cure for PTSD in general. So some, for example, physicians, they might, for example, subscribe specific, for example, treatment or drugs like anti-inhibitors, for example, or anti-stressor to be able to reduce the symptoms with those patients in particular. So it's a bit difficult to deal with something, I would say, new to the field and has no certain criteria in particular to go with in terms, of course, of like medication or treatment. So from like a public health perspective, there is a need, of course, to prevent or limit the happening or the causes of happening, for example, PTSD in general. So this is kind of like a preliminary stage or phase that we have to go with. Then if, for example, it's already there and there is no, for example, way of not having it, of course, the secondary prevention, for example, come after it, and so on.
Mm-hmm. Yeah. So another specific question, just curious about. I know sometimes in in these areas, it's with the biomarker, it's hard to, from studies even have to kind of, there's alteration, and some studies show things going up, some studies show things going down. But for chapter 2, I think page 29, you mentioned like there is interest in neuropeptide Y from 2,000 healthy US Army soldiers, and these soldiers participate in survival course, which was designed to stimulate the condition experienced by prisoners of war, so stressful experience, and the neuropeptide Y went up, increased. But also soldiers who were exposed to resilience training, they were trained to have better resilience, also have increased in neuropeptide Y. And I was just reading that, I was wondering why both went up in the same direction. I know the field might not know, but what would be your speculations?
Could you be very short? Could it be a very short answer, please? We're running out of time.
Yeah. Of course, it depends, because here it says also like the levels, of course, went up within a few hours after being exposed to military, for example, during their survival course. So this supports, of course, the idea of having, or that in neuropeptide Y enhanced stress resilient, which is, for example, generally seen, of course, in, for example, humans, such as the two groups that you mentioned. So the preclinical and, of course, clinical study corroborate these, for example, data suggesting by the reduce of MBY in the central nervous system associated with PTSD. So this is the most common SNP studies, that the MBY actually associated with the low concentration of resulting in hyperarousal of the
Thank you, Mr. Jowaf. Thank you. I'll give the floor back to Mr. Prorector.
Thank you very much, Professor Youssef. Then we now come to the following opponent, who's also a member of the assessment committee. That's Professor Soubry. She's professor of epigenetics and working at the lab of epigenetic epidemiology in the Department of Human Genetics in the Catholic University of Leuven. And thank you for coming all the way from Leuven.
Thank you, and thank you to the candidates for your clear presentation today. I will be very brief. My first question is related to the harmonization of your findings in several chapters, and I will ask about one example, the ring finger 39. In mice, chapter 4, you found several important genes, in chapter 3 in the Prismo study, and you finally ended with some biomarkers in chapter 6. So when I come back to the RNF 39, how does this fit in the story of your other chapters? So far, I didn't see this coming back into the findings of up or down regulated methylated promoters of these genes in the blood samples of men 6 months after trauma, if I'm correct. So I'm wondering, where can I find this gene in the chapter of the humans? Or maybe I missed it, or can you comment on this?
Yeah, highly esteemed opponent, thank you for your compliment, and of course for your question. Of course, there was some previous studies related to RNF 39 and also DUS22, which lead, or they kind of like associate with PTSD in particular. And I wasn't, of course, able to fully include some of those, but our team had kind of like a previous investigation in previous studies, and they came across, of course, different genes, such as, for example, RN39. And for us, it was kind of like a follow-up, but probably maybe without fully linking this particular, for example, gene in particular with the previous findings from previous studies that weren't involved, or like I weren't involved in particular in my thesis.
Okay, a second question, a general question about throughout the thesis, you measured, for instance, 6 months after trauma in human and I don't know how many months in the animals. What about long-term effects, several years after trauma in human, or offspring effects even in mice? Did you or colleagues of you or the literature, what do you know? How do you think about, or what is known about longer term effects? Can you comment on that?
Yeah, absolutely. First of all, in relation to our studies in particular, we had investigated the long-term effect and not the short-term effect in particular. So despite that, the study that we made in particular for the Prismo study or for the RNF39 study or the staining one, we focused on the long-term effect. For the Prismo study in particular, we have 5 different time points, but keep in mind that PTSD person or patient in particular, you cannot easily drag any information or or know that much about having PTSD or not unless you already exceeded a certain months. So that's why it was our focus in the Prismo study in particular to look into the soldiers that they went to the war zones or to impact zone, of course, after 4 months after the deployment, because they also went through different stages in terms of training or academic stages. And we came across, of course, the 6 months as a time point in particular. So our investigation was really into that, but keep in mind that we do have different also samples like the baseline and 1 month after, of course, deployment and 6 months after deployment, but there wasn't, for example, a possibility to dig also or to know more about those in particular, of course, for the sake of time and also for certain limitation, like, for example, the baseline in particular, we came across an issue, for example, with the quality control of our samples that we had. So we kind of exclude them, and we thought since our main focus will be on the long-term effect, so let's see the 6 months in particular.
Yeah, but you finally concluded that, and you made a nice mediation model based on SNPs. So my final question is related to that. So you could potentially select soldiers based on having a chance to be more or less resilient. So in the future, that means you would be able to select for these kind of people that are more resilient to go to war. So can you comment on this potential consequence, or what do ethicists think about this?
Short answer, please.
Yeah, of course, keep in mind the main focus was on knowing or dividing people, or dividing our sample group to 2 groups, like susceptible and resilient. In terms of the resilient, they are really always similar, they are similar to the control group in particular. But in our case, actually, the resilient and the susceptible group, they both were to for example, to the academia or the military academic courses and so on. So the resilient group, it's acting similarly to a control group in this particular, but of course, our focus was on the susceptible one and how can they be distinguished.
Yeah, okay.
Thank you very much for your opposition, Professor Soubi. Then we come to the following opponent, that's Dr. Klabbers. She's director of the Master Program in Global Health in the Department of Health, Ethics and Society at the University of Maastricht. Dr. Klabbers.
Thank you, Mr. Prorector. Dear candidate, first of all, I would like to congratulate you on the publication of this highly relevant work. My congratulations naturally extend to the supervision committee as well as to your family and friends. Now, you have made a substantial contribution to unraveling the complexity surrounding PTSD, a condition seriously impacting quality of life for those affected and for their social surroundings as well. So you have shed light on the factors involved in onset and progression of the disease and on the biomarkers that indicate the condition or mark the vulnerability towards the condition. For if we want to successfully intervene or prevent PTSD, you're opening some of the contents of the black box that PTSD is definitely provides relevant input. However, when reading your work, it also raised some questions in me. Now, I hope you can shed some light on the following questions or considerations that I have, and they predominantly concern chapters 5 and 6. And in specific, the theoretical stance. And this first question also relates to the question that Professor Yusuf has asked you. So this first question is about the problem definition. In chapters 2, 3, and 4, you examine molecular and epigenetic changes, stress modulators, and transcriptomic differentials in PTSD, which are all very much individual biomedical understandings and approaches to health and disease. So if we find anomalies in the body, then we can target those and fix the bodies, right? However, in chapter 5, you start highlighting the public health side to the problem, and you synthesize evidence on public health measures that can be taken, supported by the social ecological model. Now, that approach is fundamentally different for public health targets population groups instead of individuals, and furthermore takes the social, economic, and political environment of the affected people into account. So individual versus public health approaches lead to very different implications for interventions. Now, my first question is about what you consider most important. What should our main objective be when it comes to intervening on PTSD? Should we fix the individual body? Should we fix a population group? Or should we maybe try to fix society, and why? In other words, if I were to give you a bag of money, a substantial bag of money, what would the first thing be that you would spend it on? Your opinion, please.
Esteemed opponent, thank you for your compliment and of course for raising up such a nice question. Yeah, frankly speaking, as you know, like my thesis in particular has a combination of different perspectives. So as or from a public health perspective, it's not easy to eliminate any of the, for example, the socio-ecological model in particular. That is because actually the starting is the individual, but also this individual is having relationships with such families and so on. And this lead, of course, to having the community and also to the society. And this takes us to the social or societal level as well. So in each individual or each level that we had from those four, of course, we have to try our best as a public health practitioner or like from the public health perspective to have different preventions in each level. So that's why we do have the primary and the secondary and the tertiary preventions for each of those four levels. So I don't think so there is a possibility to eliminate any of the levels in particular, because this is like a chain. You cannot drive somebody or take somebody or take such or any level from those away and keep going, because it's like a loop. I believe the starting might be like having the primary prevention directed to the individual. And if, for example, if this doesn't work, we could go and escalate it to the next level and so on.
And how would you then do that? Use that money to target or to use it for primary prevention on the individual level? How?
Thank you, Dr. Klabbers, for your opposition. Mr. Candidate, you may give a very short answer. You might also say enough is enough.
Yeah, of course, it's not an easy task to be done in terms of working only on an individual, but also we have to keep in mind working together with individuals and, of course, the society and the community. There might be a possibility, of course, to treat them and to interfere between those in a nice, of course, perspective or way.
Thank you very much. And thank you, Dr. Klabbers, for your opposition. Yeah, Mr. Candidate, Mr. Al Jowf, the time appointed for the defense of your thesis has now passed, and the degree committee will now withdraw to discuss the quality of your thesis and the way you defended it. And I ask you and your company to await the results of our deliberation and our return. And with having said that, I suspend the meeting. So I reopen this academic ceremony, and Mr. Al Jowf, the degree committee here present online, has assessed the quality of your thesis and the way you defended it. And in view of its positive verdict and taking into account your previous qualification, the degree committee has decided to grant you the degree of doctor. And Professor Rutten is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Thank you, Mr. Prorector. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Good. Then, by the authority vested in us by law and in conformity with the decision of the committee here present online, and hereby confer upon you, Ghazi Ibrahim Abdurrahman Al Jowf, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university, shown by the beadle.
Thank you so much.
So shall I?
Yeah, the floor is still yours.
Okay, then I'll immediately start with the laudatio, Dr. Ghazi Ibrahim Abraham Jowf. Congratulations and compliments to you by us, the three members of your promotional team, including the formidable Laurence de Nijs, the formidable Lars Eijs. What an accomplishment. When having a look back at your trajectory, I remember that you contacted me already in 2016. You, an active person with a medical training and a great scientific interest, contacted me in the phase that you were finalizing your master's research thesis on the relationship between mental well-being and the behavior of workers in the mining industry. And that was at the Edith Cowan University in Australia. It was clear to me that you were ambitious and really interested in the field of public health, but also wanted to deepen the neurobiological understanding of mental illness. You expressed an interest in the topic of exposure to traumatic stress and have been working on this topic for the last years in our setting. Also, with a basis at your home institution at the Public Health Department at King Faisal University, you obtained a fellowship from the Ministry of Higher Education of Saudi Arabia and thereby could conduct your PhD studies in our team in Maastricht. From day one, you were inspired and supported by your mentor, Dr. Youssef Al-Mossad, who has been your mentor already for several years. In addition, you were also greatly supported by the Saudi Arabian Cultural Mission Team. You and your wife made a great personal investment to come to Maastricht, and you settled with your family here in Maastricht to do your research activities belonging to your PhD thesis. Your PhD project was highly ambitious, and you made use of several research methodologies ranging from epidemiology and public health to clinical phenotyping, molecular biology, immunohistochemistry, and system biology. And you have accomplished great things. You made significant progress. Over the last years, I've been very much impressed by your positive energy, your social competence, your altruism, your ambition, and your stamina and perseverance, which really brought you where you are today and deliver and defend successfully your PhD. In addition, you coped very well with several challenges, including frustrations and insecurities in your research, and also challenges that came up with the COVID period, and particularly having your family also in Maastricht. Chapeau. I also want to congratulate your family, especially your wife, and the three generations of Ibrahim, your father, yourself, and your son. I want to acknowledge the formidable supervision by Dr. Laurence de Nijs and Dr. Lars Eijsen, and also the other members of the team here in Maastricht. And Dr. Eijs and Dr. de Nijs have been your daily supervisors for the last years. I want to express my gratitude and my deepest respect to you and this entire team. Besides this, I would like to express our thanks to your fellowship provider, King Faisal University, and also to Dr. Al Mossad. It has been a wonderful pleasure to work with you, and I hope that we stay in close contact. With that, I would like to pass over the laudatio to Dr. Lars Eijsen.
Thank you very much, Professor Rutte. Dear Ghazi, Dr. Ghazi Al Jowf from now on. So I'm also very excited to congratulate you on your degree. And I also want to share a few words with you. As was mentioned already, we were your daily supervisors and we shared many experiences. So I want to get back some memories here to the floor. So for me, the work that was discussed for you coming over, I wasn't involved. But the first thing I was involved that we had these transcriptomics data that need to be generated and analyzed, and I was asked to supervise this. But we also already quickly realized with your public health background that we had to expand the thesis topic a bit, and also for us get a bit out of the comfort zone, right? To include domains that we commonly don't work on so much. And then you come into very different sort of challenges related to thesis. And I think also for you, you came into a rather experimental wet lab group, right? Doing all these things. This posed challenges for you, and you had to familiarize you with very new domains for you. Okay. Obviously, there was also this crisis that we faced, as we all know about, and we had to change some of the projects, and that made that you were even sent into the wet lab. And I'm really proud and to see how you picked this up, things you haven't been trained in at all originally, and that together with co-promoter Laurence de Nijs and several of our technicians, you managed to do several lab studies, lab microscopy studies, which I think is quite an achievement when you're not at all trained in this domain. Yeah, we of course met every week, sometimes a bit more, a bit less. And what I also still remember from this meeting is that you always took care of us very, very well. You're a very social person. And the first thing I think, as your co-promoter can confirm, you would always ensure we have a fresh coffee from a good place that was waiting for us to open the meeting. And commonly, we were even lucky enough that this went along with a little snack that you brought us, which I think is very, very kind and a very nice way to do the meetings. I actually think that in that restaurant, you were one of the most well-known customers, and they know you by face and by name, I suppose. But I also want to mention that you were supported very well by your wife, and not only you, also us. So we actually noticed when your family already went back to Saudi Arabia that there was a change, and that the provision of very nice Saudi snacks stopped, and we have. I really want to say this was very nice for us to be exposed to all your local traditions, also in food, and that your wife so kindly provided us with these items that we enjoyed dearly. I must also say that on many days where I didn't find time to organize my lunch, and then I look and I see I have a meeting with Ghazi. Okay, it's not a problem. I can do that. What I also learned, Ghazi, is you worked very hard and you were very commonly quite available. When I wanted a meeting at more easy times for me, at times of the day, or shall we say times of the night, you would commonly be available, and we have the meetings at untimely hours, let's say that. There was one thing that kept confusing me, and that was the time you had to collect your son. As you remember, it was different depending on the day of the week of the year, and way too complicated things for me to deal with. So I always managed to exactly propose times in that little time slot you couldn't do, and I still didn't manage in the end. So my apologies for this. Two more things to say. I think over time, we also, besides the working relationship, we also developed a bit more of a personal contact, and I also include Dr. Laurence de Nijs there. So we shared also more on the personal level several thoughts. And I want to say there that especially in the time of Corona crisis, where you would write as it affected us all, I have to say in your case, no, it was not as it affected all. It affected you more than others. Why? You were in a very strict scholarship in time, not being from Europe, and had to extend these, and this brings much more stress. So I think the topic you studied, you also experienced, and I think this was more than the average local student, and I really want to say that. And I really very proud how you managed to deal with all the extra organization load and the mental, consequences that had and the uncertainties this brought, apart from doing your thesis work. So all in all, I think we can look back at a sometimes challenging but also very rewarding time together. We can be very proud of your performance and degree, of course. I think your future will be bright with the opportunities you will be offered at your institute back in Saudi Arabia. From my side, I'm happy to have met you, to have worked with you. And I want to finalize by congratulating you again with the obtained degree, but also now including your loved ones, your parents, your wife, your son, your colleagues at King Faisal University, and your friends back home and abroad. And I say all the best, Ghazi. Congratulations.
Thanks, Lars.
Thank you, Dr. Eisen, for this nice laudation. Dear Dr. Al Jowf, also on behalf of the Maastricht University and her deans, I congratulate you with the degree you have acquired. And I, of course, hope you can celebrate it. And I would like to extend my congratulations to your wife and family and friends, and also to your supervisors, Professor Rutten, Dr. Eisen, and Dr. Nijs. I would like to thank the members of the opposition for their work, and of course the UM support team with the leading person, Luc Peters, and of course Bedel Simone today. And having said all that, I wish everybody a pleasant day, and I hereby close this academic session.